parasite.
Yeah, definitely.
That's pretty common.
Awesome. So again, for those of you just joining us, my name is Matt Holden. I am the Science Communication Fellow aboard this watch for this dive. You've also heard Steve over here talking. He is our lead for this dive, one of our co-scientists on this expedition. Just want to do a quick round of introductions. So again, my name is Matt Holden. When I'm not at sea, I am a high school science teacher in Fayetteville, Arkansas. Annette, would you mind giving us a little introduction of yourself?
Sure. I'm Annette Govindarajan, co-science lead on this cruise, heading up the eDNA sampling with our Multipuffer sampler on Hercules. And I'm a biologist at the Woods Hole Oceanographic Institution, where I study deep sea biology.
Very cool. So excited about using this amazing new technology,
Yeah.
this eDNA multipuffer. Hopefully it'll give us a nice, good picture of everything down there.
Hope so, thanks.
We're looking at here a sea star called the Brisingid. It's an order of sea stars that's similar to something you might imagine to be like a sea star, but these are a special deep sea group often, although you can find some representatives in shallow water at the higher latitudes. But you can see several arms. Each of these arms is covered with lots and lots of very sharp spines. It's one of the ways they can defend themselves or perhaps feed, but we're looking at actually the possibly on one of the arms on the left side, a wound or some sort of
Yeah, where it's
joint,
kind of bending right there.
Bent. Yep. All right, that's a good image, pilot. I think we can move on there. We got some good imagery.
Do they always have the same number of arms or legs like that?
No. I mean, sometimes they'll be missing appendages. Okay. Yeah. There are some xenophyophores too. These little look like balls of crumpled up paper on the seafloor. Those are small single-celled foraminifera animals. They make their structures out of sediment, agglutinating sediments together.
Very cool. So they kind of build their own homes.
Mm-hmm. They're very common in deep sea sediment, soft sediment environments. So I will give you a heads up front row that the bathy is a little bit off here, and we are heading up slope, but if there's any hard substrate targets on sonar, we want to kind of steer towards that to the most extent possible. And the waypoints are more like guidelines, so we'll just kind of keep the hard stuff in our, yeah.
Yeah. All right, copy that.
More like guidelines, yes. I said that a bunch of times yesterday. No one picked up on it, so thanks.
We can continue on with some of our introductions on the back row. Leela, would you mind telling us a little bit about yourself?
Of course. Hi, am I really loud?
Sounds great
cool.
to me.
Hi, I'm Leela Madhava. I am the data logger for this shift, meaning I'm jotting down important events and keeping track of samples as they happen. I'm originally from Chicago, but I am now a student at University of Seattle, University of Washington, Seattle.
Awesome. UW. Woo-hoo.
Let's go Dogs.
Mariana, would you mind telling us a little bit about yourself?
Hi, I'm Mariana. I'm from the Marshall Islands, and when I'm not at sea, I'm a student at Hilo, University of Hawaii.
Awesome.
And I'm taking my internship here in the Marshall Islands with MIMRA, so that's why I'm here.
Very cool. Is there anything specific that you are studying right now at school?
Just marine science. I haven't even thought about that yet.
Cool.
But I'm more into conservation work, So
Love that. Super important in the world we live in today to make
Yeah.
sure we're protecting all of these amazing organisms and biodiversity. And then also on the back row, we have Steve. Steve, would you tell us a little bit about where you're from, how you got involved with OET?
Sure. So I am a biologist. Otherwise, if I'm not at OC, I'm a biologist at the National Museum of Natural History in the Smithsonian Institution in Washington, D.C. My focus for the past at least 10, 12 years has been deep sea ecosystems, focusing on deep sea corals and how the ecology of deep sea corals and seamount environments. And for the past 10 years or so, I focused almost exclusively on the Central Pacific seamount environments. There's a sea pen right here if you have time to, or actually no, it's a sea cucumber. But if you have time, we could take a peek. If not, it's okay.
Very cool. Oh yeah, look at our little friend up there.
Partial is fine.
He looks translucent, similar to the one we saw in a previous dive almost.
Mm-hmm.
And so we've been seeing some corals on these dives. I know that shallow water corals have a symbiotic relationship with algae. Do these corals also use algae for photosynthesis and getting their energy sources, or is it something else?
No, they're strictly heterotrophic, as we say in the biz. So heterotrophic means that they are wholly reliant on particulate capture or animal capture from the water column.
Okay.
So they're reliant on consuming other things for their energy source.
Nice. So just like, most of the animals we see on land, heterotrophic.
Yep. Great. Good shot with that. I think that's enough. We saw that on a previous dive. It's so that is a type of sea cucumber, possibly in the genus Amperima, which is also the group that it's more of a common name, but it's called they're called sea pigs. You might have heard of that.
Is that are they related at all to nudibranchs?
No, these are so those are mollusks.
Okay.
And these are echinoderms.
Gotcha.
2 different phyla.
Very cool.
There's a lot of things in the deep ocean that look similar, gelatinous, somewhat,
Okay.
perhaps translucent, but evolutionarily quite distant.
Interesting. Yeah,
Yeah.
they have those very similar looking body as well.
Yep. What you
Big
got
old
there?
boulder.
What's that red thing in the foreground?
Oh yeah.
Is that shrimp in the foreground there? Big shrimp.
That's a huge shrimp.
Val, we got a big rock.
Now that's a rock.
Front row, would you guys mind giving us a little introduction? Dan, if you're ready, we could start with you. No problem.
Nice looking shrimp.
Yeah. And they use those.
That's really interesting shrimp. Yeah, this is a Caridian shrimp, probably in the genus Heterocarpus, but might be different. You got any more? It's in a really nice posture. That's why.
It looks like it's feeding or
Oh,
trying
actually,
to.
yeah.
I
That
just
actually
had
might not
shrimp
be.
for lunch.
That's all the zoom I got.
Oops.
We ate your relatives. So sorry.
Very cool. Okay, that's good. Great. Mm-hmm.
What are all those little hairs along its legs?
Actually, they're probably sensory.
Okay.
A lot of the things here, as you can tell, it's pretty dark if we don't bring our own light with us. So things have to survive on other sensory or other ways to sense their environment.
So, Steve, one thing I was wondering, I was going to ask if it's mostly like marine snow or sediment on the bottom here, but is there a way to distinguish it in soft-bottomed areas like this?
No. There's a, one second. There's a large tunicate right there. We're going to know what that is. So, but a snap zoom would be helpful on our way around the rock.
It's got a good spot up there.
Yeah.
That's good. Yeah, this is a tunicate. In the genus Cuniolis. We see these, they're very widespread, but it's a tunicate that lives on the on long stalk. Sometimes they can actually get quite long, and they just live by filtering water.
Is that about average size for them?
Average is difficult to say because the size is often dependent on the depth of the water,
Okay.
and there's a lot of different variables, including age. What do you, something like that.
And so for those of you following along, if you can see the two little green dots, those little lasers, those help us determine size. They are 10 centimeters apart, so they allow us to determine how big these amazing organisms are.
Bridge nav.
Yeah, another brisingid. Yeah, that's sufficient. We can keep moving.
You just tell me.
Setting
if
a good
the ship
pace.
to hold.
That's good. That's keeping the biologists in check a little bit from.
Busy over there with our videos.
All right, we can keep the introductions going. I'm Dan, I'm our video engineer. This is my first journey out here, second dive, and I'm really excited for what we're finding down here.
Yeah, super exciting stuff. ROV pilots, if you have time, would you mind giving us a little introduction?
I can go. I am Rai, and I'm in the co-pilot seat. And this is not my first time out here, but it might look like it is.
Rai's a seasoned veteran.
No.
She's been out here many times.
It's coming up on a year now of regular diving, so.
So cool.
And I'm Levi Unima, flying Hercules right now, and this is also my first time flying Herc for this cruise on my second dive, so I'm with Dan. We're same.
Awesome.
Yeah. And I'm Gabby Espino. I'm a navigator, so I'm more or less doing the situational awareness between the boat and the ROVs underneath us, making sure we're all one team heading in the same direction. And yeah, I'm originally from Kailua, Hawaii, and I'm a Navy veteran.
Oh, awesome. Thank you for your service.
Thank you for your support.
All right. Are you okay if I click onto your hydraulics page? I'm not going to click anything. I just want to see. Okay. So you've got those. I just don't. Okay.
We just had a question come in. Maybe Steve, you can help me answer this. Obviously, these organisms live down in the deep ocean where there is very little, if any, light whatsoever. Do they have the ability to sense light, or have they lost that, or maybe never developed it, or maybe they do have the ability? I don't know.
Yeah, light,
I'm just
you know,
messaging
light,
Robert.
light is a catch-all term for a lot of different types of radiation. I mean, visible light, probably not so much,
Okay.
but maybe they could see in other wavelengths that we may not be aware of. There's been some work about, you know, animals that can see in the UV spectrum. Those are possibilities, but I think there tends to be a more of a reliance on sensory appendages, sensing vibrations, pressures, things like that, from what we can see based
Very
on the morphology of some of these animals. But optics is a really interesting area that there's a lot of work ongoing
Yeah.
about how certain animals, especially crustaceans, can affect or can sense their environment.
Can they also sense changes in like different chemicals or nutrients in the water?
Yeah, that's probably one of the mechanisms they use.
Awesome. I know, I think it was mantis shrimp have really amazing optical sensory, especially for ocean critters.
We started today's dive a little bit deeper than
Step 20 meters, heading
previous
210.
dives, which could be a reason why we're not seeing as much as we saw on yesterday's dive, or day before yesterday. We're a bit deeper. We're about 500 meters deeper than when we started yesterday. And typically, the deeper you go, the less dense the biology. It becomes difficult to gain food as you get deeper in the ocean because of degradation of carbon as it sinks through the water column. In addition to corals and sponges, bacteria love to eat anything in the environment, so they're responsible for a lot of the low quality by I mean low carbon content in food that animals at this depth might encounter.
Gotcha. So do we find like a high biodiversity of bacteria normally in these deep sea environments?
Not necessarily. Diversity, that's another, yeah, microbial ecology is another thing. I don't know too much about that, but typically the deeper we go, there can be high biodiversity, but it's going to be patchier, for example. So it might be hard to sample an entire microbial community from, you know, one Niskin bottle or something like that.
Right.
And you would have to use some form of molecular tool to examine the diversity of sequences and for microbes.
Gotcha. And probably some environments or maybe some specific substrates might have a little bit more variety or diversity than others.
Sure. But the other aspect is some animals can offset the low quality food by just consuming more of it. And a lot of animals that are attached may not be able to move to a better location. However, they can settle in another location. For example, if you have high current flow in an area, that usually is beneficial for animals that are attached, so they'll preferentially settle in areas that have good flow, so they're receiving more food per unit time.
So they can kind of pick and choose where they want to settle?
Yeah, and
Awesome.
that's my working hypothesis for now, why we're not seeing as much here, is that it's probably a lower flow regime in this area. The amount of sediment on some of these surfaces, it helps indicate that too. Sediment tends to accumulate in areas where there's not a lot of disturbance. Think about your home, you know, where does dust settle, you know?
Everywhere.
It's definitely not in your, you know, in your main thoroughfares and your hallways and stuff like that.
Oh
It's
yeah.
usually behind the sofa or underneath the bed. Yeah.
Things I haven't cleaned in ages.
And then you mentioned preferential settlement in different areas. What does some of that life cycle kind of look like in terms of, are the larvae being spawned and then released into the water column?
Yeah, that's one mechanism. Different organisms have different modes of reproduction, but that's one of them that's predominant. There's a nice brachiopod. I think that's what you're going for down here in the center.
Brachiopod. What kind of organism is a brachiopod, Steve?
It's actually, yeah, let's take a look. The last watch saw one of these earlier on too.
Good catch.
Yeah.
Seeing this.
Yeah, it's like camouflaged very well with sediment.
Yeah, it looks exactly like the last one. These are small shelled animals. They could look like bivalves sometimes, but sometimes it's really hard to tell them apart. But they're typically small shelled animals, but the anatomy is a little bit different than your bivalve mollusks. They're a bit more simple.
Gotcha. So I know bivalves are common in seafood. Would people ever
Looks
eat
good, Pilot.
something like this?
This is not something you would want to eat. The shells are very brittle, and there's not a lot of viscera inside them.
Gotcha.
Yeah. We've sampled them a couple of times before, and they usually just get crushed. They don't sample well. They're very fragile.
Yeah.
But they are an ancient lineage that's reflected in the fossil record. So if you're interested, go look for fossils of brachiopods. They're all over.
Very cool. You find them all over the ocean, not just in the Pacific?
Nav.
Right. Yeah.
Gotcha.
Typically deeper, but they can be locally abundant in some places too.
I've changed my mind. Oh, sorry, I'm on SPL.
It's really great that we're able to share these experiences and this exploration live with, you know, people all around the globe. Just for instance, right now we have people in the U.S., Canada, Australia, Sweden, New Zealand, Netherlands,
Looks to be
Marshall
something over
Islands.
here. Do you have enough tether to get over in that area? Looks like a stick over there, a stalk of some type. Could be an optical illusion, but it's worth chasing down anything here.
Got to catch them all.
That looks like another brisingid. Or crinoid. Yeah, snap zoom would be enough. Crinoid.
Crinoid.
No, that'll be all right.
Could you tell us a little bit more about crinoids, what kind of organisms they are, Steve?
Yep, they're echinoderms, so that animal is very closely related to
That's sea
That
cucumber?
sea cucumber.
Awesome.
Yep. So
Something in the
Whoa.
water up there.
Yeah, something was
a small polychete. Yeah.
kind
I
of
think I saw that.
agitating up there. Very neat.
It's
Yeah.
dancing.
Oh, wow. Fast, little friends.
What's neat to think about is that we are the very first to ever see this
Yeah.
seamount.
It's so incredible being on this expedition, getting to explore these areas, you know, new to science, maybe even find some new species or new range extensions. Just absolutely amazing.
Actually, can we zoom right here? There seems to be a snail or something on, maybe.
Ship's holding.
It might be nothing, but yeah, that's nothing. Usually when you see patches that are, yeah, that's nothing. Never mind. Usually when you see patches of bare sediment, sometimes it can indicate feeding traces. And snails and some animals will clear the sediment, so you'll be able to see where they've traveled and where they've fed.
Like little trails or little paths through the sediment.
Mm-hmm. There's a
Awesome.
coral right here. Mm-hmm. This might be a collection target, as a heads up.
Looks like something over there on the right as well.
Yep. So you can plan ahead. You might want to collect a piece of this. This is
for the coral.
This is going to be a Primnoid octocoral. Well, we'll get a genus on it, and after we get a zoom.
Good for zoom.
Yeah, really nice.
Is there a
Are we
shrimp down there
feeling
too?
like
Yeah,
this
there's
is
a
going to
shrimp.
be a sample?
It's going to be a sample, yeah.
Lovely.
Is there any way to get a tight zoom on that node that's on the bottom? Yeah. So for the collection, since it's small, I think we can go ahead and snip it pretty low. Yeah, next to the rock.
Sorry, shrimp.
This is a good time to talk about bioaggregation, I guess.
Yeah. Tell us
When
about it.
in the deep ocean, a lot of animals will tend to aggregate around each other because for that shrimp, the coral is a structure, it's a habitat, it's producing fecal matter, and so other animals might feed on that, or it might attract other animals, or say a fish, gets captured by the coral and falls down to the bottom. The shrimp might be able to take advantage of that food source.
Awesome. Are there certain types of organisms that tend to be associated with these octocorals?
Oh yeah, brittle stars, for sure. I'm trying to figure out what some of these small animals are. They might be small crustaceans, but we might be able to take a look at them when they get up on deck too. But this is going to be definitely a Primnoid octocoral, and I'll be able to tell you a little bit more after we collect it based on some of the morphological characters, if we can put it in a genus on the seafloor, if we have to wait until it comes up to the surface.
What sort of morphological characters do you look for when identifying species?
With the Primnoids, so you'll find and you'll see when we touch the colony, all the polyps will contract almost instantaneously, all at the same time.
I'm just gonna get the arms set up.
They
the
have a
right way.
nerve net that runs through the senenchyme, or the tissue that runs between the polyps, and so they can send signals very quickly from one end of the colony to the other. But as we touch it, all the polyps instantly close, and the way that the polyps contract and retract will tell us a little bit about what genus it could be, because that morphological character is very specific to certain genera, or it helps narrow it down. It's not entirely conclusive. But this family is exclusively deep sea, and several species have been described recently by scientists at my home institution, as well as others from this group of octocorals.
Is there a reason that we call them octocorals? You know, like, does it have to do with 8?
Yeah. So if we look closely, like on the umbellula we saw, it's very apparent, but each polyp will have 8 tentacles.
Oh.
Yeah, octo 8.
Makes sense.
And in addition, the tentacles have to be pinnate, or they have small little pinnules coming off of each of the tentacles. Well, there's a squat lobster there too.
Oh yeah. And those squat lobsters, do they live only in the deep sea as well?
No,
No.
You can find them on coral reefs.
Oh wow.
Yeah, they're actually very colorful and very fancy shapes of their shells and morphology, but there's certain genera that only live in the deep sea, for example.
Gotcha.
Okay, I'm gonna come up a bit, that way I don't have to think about it. So it looks like we have, something in the forward box?
It should just be the ring.
A rock in the starboard. We have 2 rocks, one in each of the starboard bio boxes.
Okay.
So the forward bio box, both of them are empty, or there's room in the second back on the
I think we can go in the
bio
forward box, either side for this one. There's no biology in there yet.
Sounds good. And are we wanting to, where are we wanting to snip the sample?
Close to the rock.
Okay, so take the full.
Yeah.
Okay. Levi, is the camera retracted?
So I've noticed as we've been doing these dives, we don't collect
Video, are
every
we done with
single
the zoom?
specimen we find. Is there like a specific genre or specific type
Perfect.
of organisms we're looking to collect in this
Right.
expedition?
Perfect. Okay.
Generally, anything that could represent a new species, range extension,
Try not to
something really
drag
novel for these depths or these waters, and that could be anything. There's a lot of work that's been going on in the region in recent years, where we have some baseline information about some major taxa, but as I've been finding in my research is that things that look similar to things we think we know are actually not, and sometimes the only way to really discern the two is to examine it.
Right.
Hold it in your hands, examine it, and then you have a record for all of time.
Yeah, that's amazing. And
Yeah, can
you
you
do
do
a
a
lot of
partial zoom?
genomic research as well, is that right, Steve?
Right. Yeah, that's genetics and genomics is.
Oh, sorry. I need to index,
So we can
but
look
I
at
don't
the
wanna close
DNA
it
to
on.
really see if it's a new species or not, basically.
There we go.
Yeah, that it helps. So
Yeah.
Where are we here?
the whole process we use is called integrative taxonomy these days.
Okay.
So integrative taxonomy means we look at the morphology, we look at other aspects of their biology, maybe their reproductive morphologies, even ultrastructure of certain. Can we take a zoom in the manip?
Yeah.
Can you get a sec?
Great grab, pilots.
Thank you. All right. We're stable there.
Yeah, that's great that we use those
you want a
multiple
closer zoom
sources
than
of
that, Steve?
evidence to really get the best picture that we
Yeah,
can.
can we
Do
zoom
you
in
want me to
tight?
zoom in on that? Okay.
Yeah. Do you want me to move it out to where the, I don't know if I'll be able to get the lasers.
That's okay.
from-
Yeah, that's okay. All right, so you see some of the polyps are starting to retract in some ways. This one, it's a little bit, can sometimes be inconclusive, so it could either be between Candidella or Calyptrophora, but we'll have to take a look. Sometimes they continue to contract further, but I think we're happy with that and we can stow it.
Roger.
What was
Yeah.
that second possibility, Steve?
I'll write it in the
And yeah, don't bring it
sea
all
log
the way
here.
out. Apparently they're
great.
having
Thank
issues
you.
with it. Yeah, that should be fine.
So
Yeah, that's great.
Candidella is a genus
All right.
that
Let's see if I can remember how to get
typically the polyps will
into the box.
not
Bop.
not contract against the axis, so they'll kind of stick straight out in whirls around the axis of the skeleton. And Calyptrophora is the other genus where they
And
tend
we
to
don't have a preference for which side this goes in?
either.
Nope, either is
Okay.
fine.
Roger.
They'll tend to retract with the polyp
Try not to get too much closer.
pointing up the axis, or we call polarity of the polyp sometimes.
Okay.
Very nice.
So that might help us distinguish between
Alright,
the
close the box.
two different
Yep.
species.
But
And I'll get back.
and then the
into
last thing
stowing.
is, before we take off here, can we zoom in this area? There is a crab down there.
Yeah.
And it may be a suction potential, but let's take a look at it first.
Oh
You
yeah.
can see it right there.
Hey buddy.
And
Yeah.
then Nav, could we get a pin dropped for sample 043?
Cool. So can you set up to suction that?
Yeah, are we
from-
gonna wanna?
Any idea what kind of crab that might be?
Yeah, it's called a squat lobster in the genus Munidopsis. And
Thank you, Nav.
there's been a lot of work and new species described within that genus in the Pacific recently. So it's definitely a candidate
Yeah.
for a new species.
We can also porch out when you get up if you want. That does make it easier to grab the oh. Ah, Roger. Okay.
And so these organisms that we're collecting, are they pretty numerous down here? Like we're pretty sure we're not collecting like
Yeah,
the only
I'm
one of
leaving
its
this blue
kind,
button right now. I just don't want it to
Yeah.
sag right now.
Yeah.
But
Yeah,
yeah, I'll,
the deep sea is a massive, massive place, and even though we're not seeing an abundance, they're omnipresent in the environment.
Yeah.
But it's patchy, as we call. So
Right.
there may not be 10
I'm
of
gonna
them
porch
in
out a
one
little bit.
area, but you know, there's not enough
think
food to
that's-
support 10
that's
of them
fine
in
for
one
me
area.
grabbing
Exactly. As you said earlier,
the section.
less and less numbers the deeper we go because of that
Yep.
carbon degradation.
The thing about corals, though, as you saw maybe on the previous dive,
We can. I think we can just leave the porch out for the sample, as long as that's fine with everybody's view of.
yeah, it looks
Yeah.
Yeah.
Steve,
sure.
you're an amazing spotter. Like I can't find any of these things until you point them out.
I've looked at a lot of seafloor. Maybe not as much as Megan, but I've looked at quite a bit.
Your eyes kind of get used to it, maybe.
Yeah, you start to shadows, especially.
Uh-huh.
You know, when you bring light down, you see shadows of things on the rock,
Uh-huh.
There you go.
and then you start to pick out shapes, unnatural shapes,
Yeah. Yeah.
Where is the thing that I'm grabbing?
unnatural rocky shapes.
Oh yeah, that's perfect. That's ideal.
So this thing here, it's
very
probably no
good at
bigger than
blending.
about 4 or 5 centimeters.
Yeah. I had
but
already lost it.
the perfect size to suction.
Awesome. Yeah. All right. Yeah, I'm gonna grab the.
Which suction buckets data do we have open?
All of them except for 4 and 5 are open.
Come on, shoulder.
I love it when we use
It's a
the
little bit hard
section
to grab
tool.
this section.
It's so fun. So fun to watch.
Alright, it's 5.
I'm going to turn off pumps 6 and 7 while we do this.
Turn
Okay.
off or just pause?
I think I'm going to turn them off and we'll restart with a new
I'm
set
just gonna
when we're
set up
done.
first.
Okay, so
I'm gonna-
confirming they
sorry,
are off
dropped it.
now.
Okay.
Thank you.
And Annette,
Yeah.
is there a reason we want to turn it off right now before we collect?
Alright.
Just in case
Grip
any sediment or particles gets stirred
There
up,
we
it
go.
seemed like a good time to turn them off. It was about time to
Is this a
switch
good
over
view
to the
for you
new
all?
set anyway.
Gotcha.
So,
Can we do
Just to
a
prevent
partial
like
zoom?
anything we don't want getting in there.
Copy that.
yeah, any sediments or kind of over representing this
Alright.
area.
Gotcha.
Yep.
I think I am pretty close to him from what I can see. So yeah, let's get suction up. Are we on the jar we want? Okay, and we're suction up pretty high? Yeah, okay.
Oh,
It's about to go
Alright.
for a wild ride.
get in
He does not want to go.
He's hanging on by 2 legs.
Oh! Alright, so watch him go into the
Success.
jar. Alright,
Good.
stop and
It's so funny
Yeah,
watching
perfect.
them
Alright,
get
so I'm
sucked
zooming
in
back
there.
out.
amazing.
Beautiful.
Nice
Oh
grab.
my goodness.
Alright.
It's like an alien
Both
spaceship
collections,
coming
really
down
nice.
and taking
Now,
Yeah.
could I get a pin dropped for this one as well?
Pin is dropped.
Thank you very
Alright.
much.
Yeah, great collection pilots.
I'd like to try it and let's just see. Yeah, I got it sagged again. I wonder if that's why we're using the blue button. Sorry, let me just like, I'm gonna
Sorry?
do arm exercises.
Nav,
just
the pin is 044, not 043. Thank you.
my guess.
And then ROV, which slurp jar did you end up putting it in? Thank you very much.
Sorry, I'm just seeing if I can get it to stop doing that.
Herc's just really excited that we were able to collect that amazing crustacean.
I'm gonna keep blue
The
button
Munidopsis
on then,
genus,
'cause
they can
otherwise
get quite
we're just
large.
sagging.
They can be like
Oh, wow.
big, chunky,
Yeah. 5-6
crabs,
inches or so. Yeah. Very
especially deeper.
Oh.
There's a phenomena called deep sea gigantism.
Oh.
Instead of this, it's a paradox. So as you go deeper, the food supply is
Right.
poorer, but animals tend to be larger.
Huh.
Perhaps because they have longer lives, they size up.
Have
You
time
want
to
me
get bigger.
porch in
Yeah.
for you?
That is very
It's an
interesting.
evolutionary
I would
process.
think completely the opposite. That's
Nice.
really interesting.
Well, even you were saying like at the poles that tends to emerge. It's called a process called deep water
I'm gonna give you back your
polar
starboard
emergence,
rail
where
cam.
deep waters are typically come up shallower in the Arctic and Antarctic.
Mhm.
So you find gigantic versions of animals like sea spiders, for example, in the
Uh-huh.
Antarctic that are just bigger, like orders of magnitude bigger than in the temperate oceans.
That's amazing, but also kind of my nightmare, seeing a bunch of giant sea spiders.
Yes. Yes, volume for pump 6, 199602. Pump 7, 194734. Yep.
Hey,
Nice.
Rye, would you have a chance to answer a question that just came in about the ROVs?
Yeah.
So we have a question that was just wondering, does Hercules or Atalanta have an infrared camera or any way to check for like hot spots or temperature variations that may, have lots of life forms near them?
Not currently. I'm not sure if in the past we've done an expedition where that was included. I seem to remember I wasn't on that one, but yeah, not currently. We don't.
Gotcha.
Okay, I'm turning on pumps 8 and 9.
So just kind of following up on that question, I think that's why, it's super important that we have our multi-beam sonar mapping, because it allows us to get a really good idea of the different topography of the seafloor. And we know that certain species or even certain amounts of biodiversity might be correlated with different topographic structures. So it really helps us to kind of pinpoint where we want to do our dives.
There's a pink something there. You see that?
Okay, so Nav, could we get a pin dropped for 045 and 046?
Just a quick snap zoom on that, because there was something over to the left, too, that I
Thank
also
you,
wanted
Nav.
to take a look at.
Yes.
Oh yeah, that's a beautiful color.
Sea cucumber, yeah.
Another sea cucumber.
No, that's okay. We saw this yesterday, so
Copy.
should be able to get a genus on it at least.
And then just to confirm,
there's
Annette,
a-
these will be pumps 8 and 9?
I think it might be
Yes,
slightly
that's
down
correct.
here.
Thank you.
Oh, there it is, yep.
Oh
A
wow.
large tentaculated thing.
Oh,
So Steve, a lot of these things have really bright coloration. Do you have any theories on why that might be, especially since it's so dark down there and you can't really see these colors that often?
Yeah, it's so there's a lot of hypotheses. Oh yeah, there we go.
nice.
Wow.
Yeah.
That's beautiful.
So this is a problematic group.
So pretty.
So this is something we've seen and sampled quite a bit in the Central Pacific, and it's something being worked on right now. It's actually an anemone. It's an anemone relative called Syrianthid, or a tube anemone. This one is sans tube, but if you were to peel back some of those outer tentacles, it actually has a morphological structure in the middle surrounding the mouth, a second row of tentacles that are different, morphologically different than the outer tentacles, which is unique to that particular group. So,
Do they have different functions?
no, I mean they're filled with nematocysts.
Okay.
But we've sampled this one at least a few times across different Nautilus expeditions, from Hawaii down to American Samoa and others. But we're still trying to work on this group. It's a work in progress, because normally Syrianthids or Syriantharians are tube dwelling, so they live in the sediment. They have a tube in the sediment, and their body is in the sediment, and their tentacles just pop out from the sediment surface. So this one lives on the rock surface, and maybe you can
So
start
kind
to
of a
see.
unique feature for it.
Yeah, as some of the tentacles settle, you can start to see in here.
Yes.
There's a second ring of
Uh-huh.
fine tentacles. So
This kind of looks like one of those elastic caps that people could put on their head.
yep.
And then, yeah, that'd be a cool hairdo.
Right.
All right, really nice imagery. I think we can move on.
Copy that.
I'm sure it's not long before my students start to adopt a hairdo like that.
So the frustrating thing about these anemones, I've collected them before, is that the tentacles will bail out as a stress mechanism. So the tentacles will pop off, presumably to scare or confuse predators, but they pop off, and all that's left is the center viscera, the center body of the thing. So there are defense mechanisms that are...
When it does that, it will regrow them eventually, I assume?
I think so, yeah, likely.
But it's got a, I'm sure that's an energetic cost, getting rid of those.
I'm sure, yeah.
Uh-huh.
Yep. Everything costs energy in the deep sea, and deep sea animals are often optimized for energy efficiency.
Yeah, it's hard to get energy down there,
Nice
I'm sure.
polychete there.
Wow.
Type of scale worm. Or there it is, swimming. Yep. Yeah, unfortunately, we don't have a genus yet for that purple Syriantharian. It's a work in progress, and something that's been on my list to work on for a bit, but I'm not an anemone expert, but it's certainly within my job description to sequence the DNA and see if it makes sense in the phylogeny and relationships evolutionarily with other types of Syriantharians.
Yeah, it's just so incredible. You know, that one of the reasons I love science is because you're always learning new things. You're always revising what you previously knew. It's always changing.
Yep. Well, I just, based on how much I've had to talk in the past hour, I would say there's a lot more biology that's starting to appear.
Yeah, definitely.
Because downstairs I was pretty quiet. There's not a lot going on
Yeah.
for the first few minutes of the dive, but
We got the good shift.
it shows you a little bit about how patchy seamount landscapes are. You can have expanses of sediment and then tons of rock, and then it can go back to sediment again, depending on the topography and morphology of the seamount.
Yeah, definitely.
But yeah.
Would you say like more unique topography or more mountainous topography is normally where we find more organisms?
Here, can we snap zoom on that? I think it's just some debris or rubble. Yeah.
Oh,
Good eye.
Yeah.
such a great eye.
Former sponge stock. All right, that's
Zooming
good.
back out.
And then there's another octocoral. I think it's the same species that we collected right up here on the rock top. Just a quick snap to confirm that.
Upper left there.
Nope, no, it's another stellodoric sponge. They saw one of these on the last watch.
Oh, wow. Yeah, it has a very similar structure to coral.
Yeah, there are some sponges that do a very good job of impersonating corals, but sponges were around first. So
Yeah.
maybe it's the corals that are in.
And maybe the
Impersonating
corals are
sponges.
mimicking them.
Yeah. So this is a sponge called stellodorix. These have been collected in the region quite a bit,
Copy.
but most of the species are actually known from the Aleutian archipelago. So higher latitude.
Okay.
But there definitely have been a number of collections from the Central Pacific in recent years, but not a lot of work's been done on them.
Gotcha. Is there anything that feeds on them?
Sponges are often not predated upon by things. That,
Okay.
Sponges are one of the oldest lineages of animals on Earth, and they have probably been living for 5, 6, 7, maybe even 800 million years.
Wow.
And they have developed over that time a lot of defenses that, especially chemical defenses,
Oh, okay. that
keep things a safe distance away. There's some sea stars that will eat glass sponges. Pythonaster is one of them. We see that associated with glass sponges preying on or on the animals living on the sponges.
Okay.
But we do see sponge spicules.
Yeah. So they see the sponge and they're like, oh, I'm going to have a little bite. And then, oh, here's another little snack over here hanging out on the sponge.
Yeah. It's
Ooh, what's that?
nice. Another sponge coming up. We're on the sponge train this morning.
Yeah.
Wow, that's beautiful.
It is.
Good for snap zoom.
Yep.
I'll go for the snap right now.
Wow.
Wow.
So this is a euplectellid sponge. Shrimp usually. Actually, there's a couple things going
Yeah,
on
it looks
here.
like something's inside.
So can we focus on the top? There was something up there I wanted to point out.
Steve, would you mind typing into the data logger that name, the type of sponge this
Yep.
was again?
That's the genus is Regadrella. It's in the family Euplectellidae.
Awesome. Thank you so much.
Yep. Do you get better light on it if you put the downlights on?
Downlights
Or-
are on.
They are on?
Yeah.
Okay. Well, I wanted to
good.
point out, actually right here, there's a Ctenophore, yeah. There's a benthic ctenophore that often is
Oh.
associated with these sponges.
Cool.
Yeah, you want it to tilt up. Oh, do you want it slower than that? Might have to.
Yeah. So the top of this sponge has a sieve plate here, but
Yeah, tilts
it's
up
characteristic of this family. But this kind of red gelatinous mass is one single ctenophore,
Oh, wow.
and you can see its tentacles streaming off
I
here.
can't
Yeah,
adjust your
uh-huh.
PRV
Yeah.
for you. Yeah, sorry.
And so often you'll find them in these types of sponges, you said?
Any glass sponges or any structure really. They can sometimes be on corals, but there you go. So that's benthic ctenophore, probably in the
nice.
genus Jophiella, which there's been some work done on recently by some of my colleagues at the Smithsonian Institution in Washington. Very nice.
Yeah.
But if there were more, I would consider a collection. There's just only one right there.
Right.
But typically they occur in clusters of sometimes 5, 6, 10 more. But we'll just get a good image of that and then carry on.
View and upper is pretty neat right now, too.
But really nice.
Yeah. I think upper is too, actually.
Alright.
Can I
Zoom
play
back
around
out.
this servo position, or? Just to see.
This is what
Yeah.
they will typically look like.
Oh, wow.
They're on sponges. They grow quite big, but yeah.
Very
This
cool.
is a Caulophacus sponge, but yeah, you get the idea.
Uh-huh.
I don't know if I have focusing ability.
But yeah, they typically find more than one on a sponge or
I
coral.
think I just have zoom.
If you were watching in the Palau expeditions last year, we actually saw many of them and
Oh, on
sampled
our,
them, and we've already proceeded onto sequencing many of them.
And so that ctenophore, is it like a symbiotic relationship, or is it?
It's a type of symbiosis. It's probably commensal.
Okay.
So that means that neither animal really has a negative effect from the relationship.
Right.
And one of them usually has a more positive relationship. So the ctenophore gets a really nice perch to string off its tentacles and feed in the water column, and the sponge is just hanging out and saying whatever.
Doesn't really hurt the sponge
Yeah.
at all.
I got a friend.
Yeah.
Yeah.
You mentioned sponges were some of the oldest animals that we know of. Do you know by chance what like the oldest type of sponge is?
1 second. There's something here. Can you do just snap zoom on that? Sorry, I just noticed that. It's right between your toes right now. I just saw it was pink. I wanted to
Copy. Is it that?
Oh yeah.
Yeah, so this is this is another octocoral, first of its family, and I think first of its family on this dive. This is Chrysogorgiid octocoral, family Chrysogorgiidae. There's a bit of work that's been going on in this genus now. Taxonomic descriptions of new species and revisions. Yeah, that's good. I suspect we'll see more of those. They're fairly common across this type of landscape, so
Okay.
maybe a collection for a later time, but I think we'll move on.
Cool. Zoom back out.
I love how we're already seeing so many different types of organisms, so much variety. Very interesting.
There is a possibility on that ctenophore thing I was thinking about now that if there's enough ctenophores that attach to the sponge, it could affect the sponge's ability to pump and filter water because they typically take in water through the outside and
Gotcha.
excurrent through the osculum and through the top. So potentially they could clog. So there may be some downside for the
Gotcha.
sponge.
But normally only if there's like multiple ctenophores in there.
That one looked pretty healthy.
Yeah.
The sponge. Yeah, I see that.
Is that something on the bottom over there, on kind of the bottom
Yeah, it
right,
could
bottom
be.
middle maybe?
Yeah, there's a. That's an old sponge skeleton. They often fall down the slope after they detach. If they die, they just
kind of roll down the hill.
Stuff falls downhill. Yeah.
All right. Want a snap zoom?
Yeah, let's do it because sometimes you can find some associated invertebrates live around these structures.
I think there's a shrimp.
Yeah, it looks like a little shrimp fin there.
Yep. So you can see that it actually looks quite dark because, like the rock surfaces, a lot of these stagnant structures will start to accumulate ferromanganese crust, like on the rock. So that's probably why it's darkened a bit.
You want your camera tilt?
But I think that's what we got there.
Is the ferromanganese something that's in the water that starts to accumulate, or?
Yeah, it's a natural precipitation from the water, and it's actually a process that is driven by small electric charges on the rock and mineral surface.
So opposites attract kind of idea?
Yep.
Awesome.
Okay, it's really nice.
Copy.
Well, I mean, the same kind of thing. Lots of metals, especially in contact with seawater, which is a really good conductor. Small electrical charges, will catalyze a lot of reactions in marine environments. Pilots can tell you all about what zinc does.
Yeah.
Or what it doesn't do, rather.
There you go.
Tell us, pilots. Tell us about zinc.
Yeah.
There looks to be something.
Oh yeah, actually, I'm interested in that red thing on the left. You've got the eyes for sure.
You wanna look? Yeah.
Oh wow.
That's
there
awesome.
you go.
Totally
Nice
did
spot.
not see that at all.
Ship's holding.
Oh, is it a little sea star?
It's a sea star.
Oh,
Yeah.
it's so cute.
It looks like it's hiding.
Yeah.
Yeah. This is a slime star, actually. Is a common name for the group of stars.
Slime star. Love that.
They're typical representatives of the deep sea star fauna in this area. There's a few different genera that are common, but
do they give off slime like the name may suggest?
terribly, yeah. They'll fill a whole bio box with slime.
Oh my gosh.
Yeah. The bigger ones. This one is a baby, but it's a good spot. I think it's the first slime star we've seen on the expedition. Very nice.
Yeah. Beautiful coloration.
Oh, yeah.
Oh, what?
Wow.
Whoa.
What?
He's yelling.
Yeah, so I was wondering if it would do that. So this particular family, the pterasterids, have a velum, which is kind of like a sheath of skin. I say skin, it was not really skin. Tissue. And so they open that to exchange water to maintain hydrostatic pressure within their bodies. But yeah, that happens from time to time.
That was so cool.
Yep.
That's full zoom.
Yep.
How big do they get?
They can get pretty big, like dinner plate size. Yeah.
Oh, wow.
This one's small.
Do it again, do it again.
Yep.
Yeah, all right, zooming back out.
That's a good action shot.
Yeah.
Yep. No, we've seen that crinoid a few times. We'll just jot down the note and keep going.
That specific one, he's just following us.
Now that would be impressive.
To see,
It's just really moving.
yeah,
Yeah.
super speed. Just loves Herc so much.
I still haven't seen one move yet.
There are some sea stars and sea urchins
No.
that can move comically fast.
Oh, yeah.
That
I
you
know,
wouldn't expect.
like I've seen them on
They'll-
YouTube, but
like,
not,
They have their little feet, tube
Uh-huh.
feet that are
not
just moving
with my
out.
own eyeballs.
The ones I always think are funny when I see them moving are scallops. It just
Oh,
seems like it should
yes.
be a cartoon, the way
Uh-huh.
they like bounce along.
Yes. And all their little eyeballs. So cute.
So we're continuing on through these lava flows. I'm going to play geologist now a little bit. But we're looking at basically lava flows and occasional patches of pillow basalts and things, but this is all volcanic environment with pretty heavy coats of ferromanganese crust.
Yeah, I know yesterday when we were
Yeah, that's great.
cutting open that rock sample from the previous dive, it had a fair amount of that crust on the
Good for snap
outside.
zoom or wait?
Yeah, let's do a good zoom on that. I don't know what that is.
Copy that.
And sea stars and starfish, are they the same thing?
Yes.
Okay, I thought so. Just wanted to double check.
Yeah. Yeah.
Yeah, what is that?
Ooh.
Is it a can?
Oh,
it
no.
Could be trash, yeah.
Oh. It
Has
is.
it still got
It looks
stuff in
like
it?
a Coca-Cola, the reddish color.
Yeah.
Yep.
What not to do in the ocean if you're watching.
Some kind of can, yeah.
Don't litter.
Heavily rusted.
Something.
All right, good spot. That's the first trash we've seen, I think, for the most part.
Yeah. Glad we haven't seen more. Hope we don't see any more.
There's a cup coral right here, actually. If you don't mind setting up for a suction.
Yep.
Cup coral, I'm just telling you in advance because cup corals are really hard
Yeah.
to
Yeah, I can do
ID in situ.
So good sample to
And they're
Take back
really
to the
easy.
lab.
You
Shut you
can
down to take a look.
you can kind of just knock them off, and they're on a
Oh.
pedestal. They knock them off with the suction, and then they just go right up. So cup corals are some of the deepest living stony corals, the same corals that make up shallow water reefs.
Oh, wow.
Same exact
Okay,
group,
approaching you a
and
little bit more.
some just will live as one single polyp their entire lifetime. They won't make
Oh.
multiples, and usually the single polyp corals will brood larvae, so they'll
wanna
have
see your
fully
portrait
developed
lamp?
larvae
Mhm.
that will emerge out of the corals when they reproduce.
Is the plan to slurp?
Yeah.
Suction?
Grab her.
Oh.
It says slurp on all my sheets.
Yeah, there you go.
Do any corals that you know of have
And I
like
imagine we're
parental
gonna want to do
care?
a zoom on it first too.
Not so much. I mean, they usually, no, not
Yeah,
so much.
I didn't think so. I just wanted to check.
Sometimes, though, you can find planula larvae attached to the outside of
Alright. I'm
corals,
just
and they'll
coming
hang
up
there
so
for a
that
bit, especially mushroom corals will do this
Okay.
often.
We're not in a ship move. Okay, nice.
Yep.
Oh, wow. Are those, that wider colored part, would that be part of its gut?
Yeah, a little polychete.
Ah.
Yep. Yep. So the tissue is the part on the outside, and that's just gelatinous, gelatinous tissue. And then the inside, where you see the white part, that's the skeleton portion.
Oh, okay. Skeleton.
This one, this is there's a few different, I hazard a guess, but this one is giving me Vonella, a genus called Vonella vibes, and that's what I'm going to go with for now. But
Try to grab it
we'll
like
we'll
this.
look at and put the ID on it when it gets to the lab.
Nice.
Because there's a few structures that are inside, underneath the mouth, which is right in the middle where it's pinker, more pink, that we can look at that would tell us whether it's one genus or another. But Vonella is a deep water genus that you typically find deeper than 2,000 meters, and it's widely distributed in this region.
Very interesting.
It looks remarkably similar to one we saw yesterday, and I'm kicking myself for not collecting it.
Nice, perfect.
Yeah, I've noticed a lot of these different deep sea organisms tend to have like similar morphology, similar adaptations. So I'm sure getting them back in the lab really helps in trying to determine exactly what it is.
With these cup corals, usually I try to collect them in pairs because they don't have a lot of tissue. Even though it looks like there's a lot of tissue, it's mostly water, and usually we have to take a forceps and get a tiny tentacle to extract the DNA. But to look at the...
Yeah, I'm pretty sure I'm like right there, so...
Oh.
Yeah.
Woo!
There you go.
Got him.
All right, let's watch them.
Well done.
Nice, got it.
Which jar did that go into? Thank you very much. Nav, could we get a pin drop for 047?
Yep.
My training was in fish suctioning, so...
Yeah?
Yeah.
This is nothing.
No. It's actually, I haven't used the arm in a while, so it's good to get back in. This is a new arm as of this winter.
I bet it's very difficult to suction up a fish.
It's a dance between pilot and co-pilot for sure. Yeah. Nice. Does back row have control over the lower camera's focus at all?
We should.
If it's maybe. Let's take a you want me to zoom out a bit, like past the porch.
There's something.
Is that something center of frame there, or is that an optical illusion?
It looked like something swam by or floated by. That top corner.
No. That's just zoom. Yeah. 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 It's just perpetually focused on the porch. Interesting stuff. It's hard mounted to the, but yeah, we can maybe talk about how they, yeah. Maybe? Yeah, on that.
So Rye, you said that your training with the suction was with fish. Would you mind telling us a little bit about like how you got into this field of work or this industry?
Yeah, I mean, up until a couple years ago, I had no idea really that this industry was an option. For me, my background is mechanical engineering, and I live on Vancouver Island, so I've kind of always worked in proximity to the marine industry.
Nice.
I used to work for a company called Ocean Networks Canada, which is like a science data company on Vancouver Island. And yeah, through them I did a bunch of expeditions with Coast Guard, and one out here on Nautilus, and that was kind of how I got in proximity to ROV and started asking questions about, you know, do I need to specifically have gone to school for this, or, and everybody was like, nope, you can just do it. And then yeah, it was actually on this ship when I came out with Oceanarics Canada that I started talking to the lead of the ROV department, Josh Chernoff, and was kind of like, you know, I'm interested in coming out and doing contracts and stuff. And then yeah, starting last year, I left my old job and started doing this as my main deal. So
That's awesome. Yeah, I love how there's so many different roles aboard EV Nautilus, so many different ways to get involved in ocean exploration.
And completely different ways. Like, I don't think any two people have gotten here like the same way, like even on our team with the same position. Like, nobody has the same background or the same, like, some people have, you know, 2 PhDs, and some people just did a technical program, and some people, you know, just worked in industry as kind of like tech people and stuff. So
yeah, taking advantage of those opportunities as they arise. I love it.
Yeah.
2 PhDs, that's a, wow.
Sounds hard, sounds difficult.
I struggled with one.
Yeah. I know sometimes when we do the go around, like everybody's background, I'm like, wow, I feel really like, oh.
Wow. Yeah, that sounds like not much life for a long time while working on those.
I like to tell people I capped out in the 27th grade. If you add up
27th grade.
That's great. Let's see how many did I do.
Yeah, that's nice. Can we get some screen grabs
12,
of that?
16.
Just the terrain, the topography.
Yeah.
It's terraced.
I made it to 21st
flows.
grade. I didn't make it quite as far.
Yeah. It's not because I was a poor performer. It's just...
Just tired.
Yeah, definitely. It's a lot of work.
I did see for the first time when you fill out forms, you know how they've drop-down menus about, you know, how many grades you completed. I did see one that said greater than 21 grades.
Oh my gosh.
Recently, and
wow.
21 grades. What does that even mean?
Yeah. Well, I guess while we're talking about how we got into this industry, Levi, would you mind telling us a little bit about your backstory?
Yeah, I'm Levi Unema, sitting in ROV pilot. My high school science teacher got me into this.
What? Go science teachers.
Yeah. So he was on the board of a nonprofit that does ocean exploration, and copy, go in there. And yeah, I electrical engineering background turned ocean ROV.
Awesome. Super cool. I love that your science teacher got you involved. I'm hoping to inspire my students to get involved in exploration and maybe come join the Nautilus team one day as well.
We're giving shout outs. I also want to shout out my high school science teachers because they were awesome.
That was Leela shouting out to her high school science teachers. Yeah, I think if anyone interested in getting aboard Nautilus and doing any type of this ocean exploration, there are so many different roles that we have on board. We just spoke with, you know, our ROV pilots. I myself am a high school science teacher. We have videographers and photographers, different engineers, all sorts of amazing ways that you can get involved in ocean exploration. I think just go for it, you know.
There's a couple of zoantherian.
What is this?
So it's a sponge stalk with there's a couple of zoantherian polyps on the top.
Thank you.
But it's largely dead. But you can see how the structure is important for other things.
Holding.
You said it was
Post sponge.
Yeah, nice little habitat.
So yeah, I'll write it in the sea log.
Are we still doing heading
rescuing me from having to figure out how to spell
zoantherians are.
Thank you.
Actually, can you zoom back in here on the lasers?
I'm right there with you, Leela. All these new words. I'm like, I gotta see it written.
Here, I'll touch you up. You want me to tilt up?
Let's see right here.
Oh, sorry.
Mm-hmm.
That's full zoom.
Yeah. It's really small, so I can't even tell what it is from here, but I think that's okay.
Okay.
Might be a very young coral or maybe a sponge of some type. But too small.
We'll let that friend grow to get big and strong.
But we're definitely starting to see more diversity, which was my suspicion. As we get higher up towards the top of this ridge and towards the top of the seamount, not only is the food becoming more enriched for animals in the bottom, but also the currents will be faster because as currents run over the mountain, they typically accelerate, which gives better flow conditions for things that are dependent on feeding on particulate matter.
Speaking of nav, are we still, where are we at waypoint wise? Lovely. Thank you.
Yeah, you're at the end of your tether.
So yeah, kind of speaking of our backgrounds, our videographer on board right now on watch is Dan. Dan, would you mind telling us a little bit about how you got into this industry?
Yeah, absolutely. I actually didn't get into this till halfway through a psychology degree, which is what my degree is in.
Nice.
Nice.
And then I had a mentor that encouraged me more towards engineering, and then I picked up in broadcasting and just loved it, and then worked my way up to engineer, and then from freelance to here ended up on this amazing ship, and I'm loving every bit of it.
Yeah, so great. Love having you. I think that's so great that, you know, your path wasn't just like a complete straight line, and I think it's important.
Nowhere close.
Yeah, it's important to realize that, you know, you don't have to follow this linear path. You don't have to know what you want to do in high school or college or even later in life. I myself did not follow a linear path to get aboard EV Nautilus, even to becoming a teacher. So I think it's important to just follow your passion, and when you find something you love, go for it.
Yeah, you'll never know what you'll find.
Yeah, I'm even from Arkansas, a landlocked state. Never would have imagined I'd be out here as an ocean explorer, but just goes to show you never know what life will bring.
You gotta be on SBL if you want your jokes to land. Yeah.
There's jokes?
Yeah, Levi's making jokes, but he's not on SBL, so.
Levi, share.
Depriving the rest of us of your jokes.
No, you guys aren't on the air usually. Yeah.
We can make space for some good jokes.
Conversation. Ooh, something over there under the rock. What do you think that is?
It's a good spot.
Swallow lobster?
I'm gonna say crinoid, but let's take a look.
You know, let's snap it.
Yeah, let's snap it.
Crinoidville. Oh yeah, for sure.
Nice.
Mm-hmm.
Good spot.
We often get a lot of these crinoids if we sample other things like corals because they also like high points in the topography, and corals tend to grow up high and branch. But the problem is that the same thing with sea stars, sometimes when they're stressed, their arms will just pop off.
Oh yeah, it looks like.
So they lose arms and they regrow them. So you'll see two different pigments of arms on there. These are known as comatulids, which is a group of crinoids. There's two major groups. There's the comatulids, and then there's the stalked crinoids, which adhere to the bottom. They're stuck to the bottom.
And these are types of coral? Is that, no.
Huh?
The crinoid, what kind of organism
It's
is
an
a crinoid?
echinoderm.
Echinoderm, okay, thank you.
So within the echinodermata, you have several different classes. So
Mhm.
you have echinoids, which are urchins. You have asteroids. Asteroids, which are sea stars. You have crinoids, which are these, the sea lilies and
Yeah.
feather stars. And then you have the holothroids, which are the sea cucumbers.
Very cool.
Ship's coming to an end of its movement.
There's something floating here. Do you see that? Oh, all right. Yep, okay. Never mind. Mm-hmm.
Schmutz.
The technical term.
So Gabby,
Yes.
you said earlier you were in the Navy. Were you a navigator? Is that how you kind of got into this field?
I was a navigator, but on aircraft.
Oh, okay.
So I flew for the Navy.
Amazing.
Yeah, so if you know Top Gun and Goose,
Yeah.
that was Goose.
Heck yeah, that's so amazing.
Yeah, except I'm still alive. So.
We're glad for that.
Yeah. That was 9 years of my life.
Wow.
And I just wanted to get back to the ocean, and ocean is my home.
Love it.
Traded my wings for flippers.
Amazing.
How fast did you go? Is that classified? What was your max velocity?
It was a big Boeing 707 aircraft.
Okay.
So, yeah, we mainly did a lot of orbits and stuff to communicate with like submarines and stuff like that.
Very interesting.
Yeah.
That's awesome.
I avoided boats my whole Navy career,
Must be
and now I
fun.
choose to be on a boat.
Interesting how it goes.
Yeah.
It's just too much fun to be out here on EV Nautilus, getting to see all these amazing environments.
There might be something here. I don't know what.
Yeah.
Discoloration on the rock.
Roger. If you're watching from home and you think Steve sounds like Matt Damon, please drop a comment.
He looks like him too, right?
Maybe do one of those polls on Instagram and have him do some lines, because I think he sounds exactly like Matt Damon.
There is something over here, though. That doesn't look like anything, but there is something over here.
Big stock.
Big stocky thing.
Changing the
agree.
subject to
I've never heard this
Once you
before.
think about it, like, you can't not
hide
hear
the fact that you're secretly Matt Damon, who's
it. He's
decided
related. He's for sure related.
Distantly,
He's
sure.
being, like,
get into ocean
low-key
exploration.
about it.
Evolutionarily related, I guess.
Common ancestor.
Definitely.
Pull some interstellar quotes or Martian quotes.
Right.
And we'll,
I
like,
haven't seen The
listen to
Martian
it on
yet.
YouTube, and then
We
it's
just watched
great.
it during the transit.
I haven't watched it end to end. Bits and pieces.
so this is another anemone that's
Yep.
taken hold on a old stock.
Yep, another same kind of sponge stalk anemone. This one, though, has ophiuroid brittle stars on the lower part of it.
Those brittle stars are wrapped up tight, aren't they?
They are.
Oh, I
Wow.
didn't even realize that's what
And
those
we haven't
were.
seen that many, which is really bizarre because usually they're everywhere.
Good for a bit of zoom.
Going for a landing. And
Oh.
There it is.
Ah,
It's
cool.
closing up.
it's like right when I bumped the ground, it
I'm
bumped
gonna
it.
slow
Yep.
you down.
Yeah, you want to set me down? Yeah.
It really does look like a Venus flytrap.
Yep.
It does.
Exactly.
So
And,
this is an anemone. An anemone.
Zoom in on the center of it if you can. And then tilt
An
me out.
anemone. Oh my God! Living on a sponge stalk with a brittle star also living on the sponge
hold
stalk.
that.
There you go.
yes, several of the
Wow.
stars. Yeah.
Come on, come on. Yeah.
Oh,
And-
beautiful.
Oh gosh.
Hi.
Are those
Nice shot.
on the brittle star? Those little appendages like sticking off? Are those its feet?
On the brittle star is the one underneath. And
Right.
Yeah, those are. So they have arms, and then these arms have spines.
Okay, so they're like little spines.
They're
Okay.
actually diagnostic for this genus, which is probably they're in the family Ophiocanthidae. So it could either be Ophiocantha or Ophioplanthaca. Both of them
Do you want me to pan down the stalk?
have very minute differences
Sure.
in the oral disc, but something you'd have
Should we
to
get in
look
the
at.
ship
with
moving?
close
Yeah,
look.
We're pretty
definitely
stretched
looks
out.
like a no touchy.
Yeah, definitely
That's it, yeah.
very spiny.
Yeah. Wow.
Okay.
But you can see that some of them have lost arms but have regrown them.
Bring it back up.
Yeah, that one,
Really
like the fourth
fast.
one from the bottom, you could see the new arm kind of coming out.
Yep.
Opportunistic.
Sometimes on these anemones you can actually see polychete worms. They live on the oral disc, but this one has none associated with it. I'm assuming they're not prey, but they're commensal. They're living in a symbiosis of sorts
Right.
with the
Collecting food that,
anemone.
yeah, makes its way in there.
All right. Looks good here.
Copy. It sounds like
Very
we're clear
nice.
here.
Open close
cool.
action.
We'll keep moving.
Mm-hmm.
All right.
That was a beautiful zoom out, Dan.
Thank you.
220.
Yeah. Yep. Let's do another 20 meters. That sounds really good.
Copy.
And I'll kind of get back center line and
Get back.
Bridge nav.
You're off in the cabbage.
We step 20 meters, heading 220.
So Annette, I was wondering if you could tell us a little bit about how you got into this field of ocean exploration.
Oh, okay. Well, I got into marine science, I guess, from my experiences growing up. I had a trip, and my middle school teachers took us to Bermuda, to the station there. Yeah, it was a great
Wow.
experience.
I wish I had that in middle school.
Yeah, I'm from Connecticut, as is Steve, right? And I went to University of Connecticut for my undergraduate, majored in biology, and then I did my master's degree in oceanography, also at UConn, and then I got my PhD at the MIT WHOI, Woods-Hole Oceanographic Joint Program.
That's amazing.
Yep.
And you're,
Disregard
you're
pilot,
with
that's nothing.
Okay.
WHOI still, correct?
Yes.
Awesome.
Yep. Now I have my own lab.
You going for that shrimp
I
one?
did my thesis work on hydrozoans, which is a kind of cnidarian.
Okay.
So we've talked about some of the jellies, and so they have complex life cycles where they have hydroid or polyp stages, and then they have medusa stages, and then
Oh.
there's a lot of variation where different species has lost or one of the stage or the other, so they may be only have the hydroids, or maybe they lack medusae altogether,
Okay.
or have some kind of intermediate type. And I looked at the evolutionary relationships between the different groups to try
Yeah.
to sort that out.
Sorry.
Did you
Mhm.
find that some maybe that don't have the medusa, or maybe do specialize in different environments, or
Well-
specialize in different food or anything like that?
What I found was that it was actually a pretty common transition to lose the medusa stage. That was considered what we call the ancestral state.
Okay.
A lot of but sorting out things at the species level is more complicated, because a lot of the species were nominally what we call cosmopolitan, meaning that they're found all over the place.
Right.
But then when you start looking at the level of the genetic level, you start to think that maybe they're complexes of species that actually they look alike but genetically are distinct.
Okay.
And so it's an issue that we find in a lot of different animal groups, actually.
Yeah.
These are what we call cryptic species that,
Okay.
that we think they look alike, but then when we start doing the genetics, we start seeing that they actually are different.
Very interesting.
Mm-hmm.
Yeah, another reason it's important to have those multiple lines of evidence, for sure.
Yeah, so like Steve was talking about, that integrative taxonomy is important.
Yeah,
Yeah, I'm confused by what I'm
very
looking at.
interesting.
Looks like a holy rock with sand in it.
It's a hole in a rock. Yeah. Maybe something in the hole.
It could be a home for something.
Yeah. Maybe something hanging down there.
How would
Yeah.
a hole like that form? Yeah.
What is going on?
Maybe
It's almost like manganese formed, and then like whatever formed around it is now gone or something. I don't know.
Yeah, that's a lot of sediment to accumulate in a hole.
Yeah. It looks like something up on the left side of the rock, too.
No, they have
Right.
there's
And on the
actually a
ground
coral,
on the
a
right.
coral branch on the right side too.
Roger. Coral. Oh, look at that. I didn't even realize this big dead.
Oh, wow.
How did I miss
Wow.
that? Nice.
Is it a
Oh, there's
sponge?
something
so
in the
cool.
hole.
Oh, yeah.
Huh.
The sponge.
There's another coral inside.
Flash your
though.
rubber cam. Yeah.
Yeah. Pretty short. I can float it. Huh.
Yeah,
Interesting.
I haven't seen that
Is
before.
that a
Let
sponge
me know
in
when
there?
you want to snap
Okay.
zoom.
Can we zoom on the hole and then the coral to the right of it?
Yeah, you can zoom in, partial. I'll try to float you back down to that
Okay.
coral on the right. Bring her back down here.
Looks like it might be a sponge in there.
Okay, yeah.
It is a really choice
Oh.
spot for a sponge.
Huh.
Yeah, it looks like maybe a farreid sponge
I'm gonna
on
tilt
the
you up
inside.
a
Doesn't
bit.
look very healthy though. And then this is a bamboo coral in the foreground, but it's very, very small.
Inside that rock, I would think it would be difficult
Copy.
to catch, to filter feed with the current.
Yeah,
Seems like a barrier.
there's a balance in where
zoom in more
some
on this
things
little
choose to
coral? I think
settle.
You want me to tilt you around?
Right here is good, I think, for now. I'm going to try to zoom in on this.
This is a bamboo coral,
Oh, yeah, tilt me up.
similar to one we collected yesterday, but this one looks like it's a branching bamboo, but it's so small. There's not a lot of morphological characters in colonies that are so small that we can really grapple onto for ID. So the one aspect of settling in a hole, some other corals do this too, is that even small changes in flow, like in a hole or around the edges of a hole, can accelerate water and food. So sometimes you
Okay.
end up with things settle
Slow
there.
you down.
It's also that potentially it could protect them from other debris that might fall down periodically,
Right.
especially if they're long living.
Steve, I have a question.
Mhm.
Why do we always see when we see the corals, they're pretty large, like you said, this is one of the smaller ones, but still we see it's bigger than 10 centimeters just from those green dots.
Yeah.
Can we see the babies? Are we just missing, or not, that babies is maybe the wrong word, but the really young ones?
The newly
Or do we
settled,
just,
yeah.
newly settled recruits, do we just don't spot those as we do our surveys, or are they, maybe the juveniles have very high, fast growth rates, so they don't exist as small for very often, or is there any idea
you're
at
good.
all
Yeah.
about that?
Yeah, so usually these corals will settle as a single polyp or a pair of polyps, and they're just too small. You usually find them, they're a half of a centimeter, something difficult to pick out. Some sites, I'd like to gauge kind of the reproductive potential of a habitat. I look at the size distribution of different colonies, and there's not a lot here to go on. We're looking at single points, single individuals. But if there's a healthy distribution of sizes of of colonies or sponges, or you can gauge
If I
that there's
zoom out on
probably
my
a
main camera, ROV.
reasonably good recruitment.
Yeah.
But
Thank you.
where I often see them is when we sample rocks. You'll often pick up individual polyps on rocks, but it's not something you
I
can see
see.
on camera here.
Okay. Okay.
Not usually.
That makes sense.
Yeah.
Can you tell sometimes when you get the corals if they have eggs or might be brooding planula? Yeah.
Yep. Yeah,
Tell me when on
they'll
Atalanta.
be engorged usually with, they'll puff up, look like, big. They have big pillows at the bases of them.
Yep.
Yeah, that's characteristic.
No problem.
Yeah, one of the things about reproduction, especially in areas that don't have strong seasonal dynamics, is that we don't think that they reproduce annually, like something that has strong seasonal signals in the environment. So these might actually only reproduce once every several years.
Right.
But a lot of the prevailing thoughts is that the
Pretty cool terrain.
corals and sponges and animals in the deep sea that are suspension feeders are triggered by pulses of food that drift down over time. So every, say, spring, usually you have a food pulse. Wow,
Look at
that is a
this
very
overhang.
nice.
Yeah. Little overhang.
That's
Wow.
that's a lava flow though. Yeah. It's
Wow.
maybe crust on most of it, but it looks like part of a lava flow at one point in
Part of me
time.
wants to like park on it and see if it collapses, but
Probably not ideal.
So you're saying that those corals may reproduce like when there is plentiful nutrients or something like that?
Yeah, well, I mean, so here's the thing about seasonality is that
oh,
in the higher latitudes, you have a very thick surface mix layer.
Ooh.
But here,
Go for it.
the surface mix layer is 50,
gotta be-
100 meters
My
at
goodness,
most.
it's big.
it's like a tractor beam.
And so what happens is it takes a
Well,
long
there's a
time
couple
for
right
food
next
that's
to each other.
produced in the shallows, which is normally lower abundance because it's not as there's not a lot of large
Fish too.
planktonic organisms like
There's a fish.
diatoms that you find in shallow waters in the higher latitudes of the fish.
I love diatoms. They're
Let's go for
so
fish.
beautiful.
The
They make
stars
so
talking
many
about. You wanna
amazing,
zoom in on the
beautiful,
fish?
morphological
Oh, there's a
features.
stock thing. Oh, there's all sorts of
Oh,
good
is
stuff
it,
here.
is that a fish?
Yeah,
Snaps in,
cool.
go in, zoom this fish, yeah.
I
Try to
love
get the-
it.
This is I think this is a grenadier or macreurid fish. Rat tail fish, sometimes they're called.
Wow. Yeah, I can totally see why they'd be called that with
Yeah.
their, looks like a rat tail hairstyle almost.
Yeah. Yeah. I can see that. Looks like a rat tail.
There's a lot of different common names, but the family for these is Macreuriddae. And typically you'll see that long dorsal fin, kind of looks like a giant sail, is characteristic of this family. They're actually related to cods.
All right, we'll let it go video. You can zoom back out. I'm gonna go
Has
for the
any,
big
you know of anyone that's ever ate
star to
one?
the right.
Yep.
And then there's a
Yeah.
Yeah?
What I missed that.
I said, do you know anyone that's ever eaten one? I know that we
Yeah.
eat cods.
Actually, grenadier is actually commercially fished in a
Yeah.
lot of places.
Really?
Yeah,
I had no
big
idea.
ones, yeah.
Right
If
here.
you have, or have ever eaten surimi sticks, grenadier is a very common blend fish to go into artificial crab meat, yeah.
I have probably eaten one then.
Yeah.
Yeah, you can take a little more zoom, yeah.
Not here,
Some
though.
of the
Usually they're
grad
higher
students
latitude
eat some.
fishes.
Yeah. Do they get bigger than that one?
Oh, they get huge.
They get huge,
They get
okay.
huge, yeah.
Do you need a closer look at this, Steve, or should
Nope.
we keep moving?
This is okay, yeah, we've seen these before.
All right.
So we
Thought
got
I
it.
saw
You
something
can see
good.
side by side crinoid brisingid, the difference
Yeah,
between
that's
these
pretty
two
cool.
here.
They're very
Two
pretty.
different classes of echinoderms.
All right. That one those look healthy. That's a big crinoid. Each leg was like over 10. All right, I saw something stocked over here. I'm gonna go for next. Actually, 2 stocked items.
So Steve, another question for you. Put you on the spot
Yeah,
here. You know, I'm just mentioning that I was,
Roger.
I did my PhD thesis on hydrozoans. Do you see many hydroids down here?
Oh, tons.
We
yeah.
got a lot of stock
Yeah.
stuff
Tons.
going.
Wow.
Like what kinds?
To be honest, I don't know the finer taxonomy, but
Is your camera still centered up on the
anything,
porch, or-
any bare surface,
I don't know.
like
You want me to
especially
check?
Sure.
coral or sponge,
take me a minute.
stalks, will have hydroids usually attached. But I'm not familiar with the finer taxonomy of a lot of hydroids, but
Yeah.
some of the
They're a tough group.
yeah, some of the big ones we see that are often mistaken for larger corals. We see
Maybe
like
it's extended
Yeah,
Solandaria
it looks good.
Mhm.
forms
Alright,
big
tilting back
branchy
up.
things in the shallower depths.
There you
I think
go.
this might
Do you
be
want,
a precious
what's
coral.
your top cameras?
Yeah,
Let's take a
I'm
look.
gonna try to go for that. Tilt down.
These
as I
these
come up.
corals,
Okay.
if it is a precious
Tilt
coral,
down.
they love overhangs.
Yeah, just lose the
Did you say
cameras,
precious
Yeah.
coral?
Precious coral, yeah.
Yeah.
So
Alright,
precious.
let me zoom
My
in on
precious.
this. Yeah. It's just like a bubble gum or something. Call these. It's kind of under an overhang, Steve.
Yeah, no, I just
It's a
wanted to
little
get an
difficult for me to
observation.
get to, but if you got more zoom, you can take it. I'll try to keep it unfloating.
Yep, so this is, I think, first observation of the cruise of a precious coral. This is called hemicorallium. It's in a really tough spot. They love overhangs.
Flush that out,
So sometimes, actually, this particular family
Okay.
gives the corals its namesake, originally described from Corallium rubrum in the Mediterranean. So the word coral came from that species name,
Huh.
and so this is the same family as that. So this
Wow.
is the first coral was not a stony coral. It was something that looks like this.
That's really interesting. Yeah, I love how we're starting to see more and more
Alright,
as we
I'm gonna
get
go up to the
kind of
top.
higher up the slope. We're at 2,444 meters right now, and we started just below
Oh, yeah.
2,700 meters, I believe.
Oh, there's a
What did you say
coral
we're
off
at?
to the left here.
2,444.
Okay. Oh, I love these sponges.
Yeah.
Which
I
one
love
you
how
want,
yellow
Steve?
it is, and it looks like a little
Well,
cloud
actually,
sitting
yeah.
on top.
Could-
Sorry, what was that?
There's something off to the lower left here too.
Yeah, this is another one of those stellodoric sponges, but we have some of these structures here, these little,
Okay.
I think these are kebab sponges. We'll take a look. So yeah, we've got some large Bolosoma sponges
Hey,
here
Steve.
in the background. Yep.
And maybe let's do a Niskin bottle here, since
Yeah,
there's
sure.
quite a variety, and we've only done one Niskin sample so
Whoa,
far.
look at that
Look at
coral
Mhm.
underneath. See
Yeah,
that?
the huge one on
Underneath
the right.
Oh,
Can we,
wow,
can we
yeah.
do a
Whoa.
Niskin?
Yeah,
Yeah, we can do a Niskin.
Yeah.
Thank you.
I'm gonna hold
that
it.
is that's a big thing in that overhang there.
Yeah.
Uh,
I'm gonna land here.
'Kay.
And then we'll look at that. Tilt me down on the mic
Is
camera.
that
Yeah,
a precious
right.
coral too?
maybe. We'll have to take a look.
I'm gonna look for
It
something
doesn't strike me
small
as one
on the ground here
because the
first.
branches look a bit thicker, but
Want
we'll
me to
take a
keep
look.
going?
Yeah,
the
keep
best
going
we
down.
can. This is a
I think
good.
you might be resting on that arm. Do you want me to pick up the...
Is the shoulder all the way up?
Yep. Shoulders all the way up. You want me to pick up the,
You sure? Yeah, just get out of the way.
forgetting names for joints. Alright.
Are we doing a particular bottle? What bottle?
We've had one fired so far, but everything else is free.
I think we've only fired one. Is that what she said?
Yeah, we've
Alright.
just done one.
So
So
we
if you want to
could-
do two?
We can fire the second one. She has the arm out anyway, so yeah.
Perfect. Okay.
Copilot, you want to fire the second Niskin? I'll float up off the wire here.
Yeah, are you able to tilt my camera as
Yeah.
well? Alright,
Yeah,
as long as you can do that, then.
I'll get up off here.
Alright, I'm gonna start bringing the arm around, try
Okay.
not to hit any
I'll back you away.
precious corals.
Alright. So I retract my camera here. And then look at
Yeah.
our rings here.
You're all the way retracted?
Yeah. Alright, so we're going for the second one.
Yeah,
Which is,
second one.
Looks like we ripped the ring off of the top one already today.
Did we? Okay.
Yeah.
So we're going for... Can we iris up? You might have to iris up a lot. Yeah.
So we're starting
this
from
one.
the top today.
I'm trying to...
She's gonna grab that one. And we got the other camera right here so we can see
Yeah,
him.
is anybody watching that? See if I can...
Yep, we're watching.
Did it go?
I don't know.
I think if I pull much more, it's gonna break.
Okay. Would that have,
I
let's
didn't see it.
That's the second one from the top. Which one, does that close one on this end or close, I think it closes one on the forward.
So that's looking from the, so it should be near the front of the vehicle. So that's looking from the aft. So near the bottom of the screen.
Yeah, I for sure see one of them close.
Yeah.
I'm not sure if that's the first or the second bottle. You know what I mean?
Yeah.
It's hard to tell.
It's really tough to tell.
It's really tough to tell, yeah.
I think we should move that camera back to the forward
Yeah,
facing.
I like the looking at the tops of the bottles too.
Yeah, same.
You want to do another one, or what's your vibe?
So-
you want to do another one just to
Yeah, just in case.
just in
Yeah.
case?
But
Yeah.
we
I think
think the Niskin fired just now, just to be
So
clear.
you think,
We're not sure.
you're not
Okay.
sure that 2 fired,
Yeah.
but you're gonna
Um,
pull 3, is that?
Is that, so that's your option. We're not 100% sure that Niskin 2 fired.
I would do
we
it.
should do another
I would
one.
do it. I
Just
mean,
in
we've
case.
just in, we've only done 1 so far, so
Okay.
I think, and also having duplicates will be a good thing.
We're gonna
anyway,
keep that
so.
sample
Yeah.
number
I'm gonna
and
try
just
to
roll
grab
to the
this
next
one.
one for the next bottle.
Keep
Yeah.
the sample number?
Yeah,
Okay.
that's a good sample.
All right, wait, keep the sample number for
Yes. So
spot
I'm
for number
doing
2.
now, that was 048, and I'm now doing 049
Okay.
for Niskin 3.
Yeah.
Okay.
Perfect.
I'm pulling this one. Everybody watch that camera.
Watching.
Okay.
Okay. Yeah,
Boom.
that one went.
Yeah.
Yeah.
I think I see.
Oh,
They both went.
Yeah.
Yeah, I think I
You're
see all
good.
3.
Yeah.
Nice.
Watch your upper cam.
Yeah.
Good.
So those of you that are watching, this is one of the methods that we use
Data,
to
it's
sample
included.
for environmental DNA.
Yeah, so
Alright.
we've got the autonomous
Nav, it
sampler
looks like you've
running in
got
the background,
2
which is giving
048s
us
there.
a
Beauty.
integrated
048
view
and
over the whole
049, that's
survey.
right.
This Niskin bottle will
Thank
give
you,
us
Nav.
a specific local point source for this particular location, which we selected just because it has a lot of diversity.
Yeah.
But it'll reflect this particular location, whereas our other auto
I'm
samplers
gonna try
will
blue button
be
again just now, just see if it.
Kind of like the whole dive, or?
not the whole dive, but we are dividing it up into 8 segments.
Oh, okay.
Right. So,
Alright.
so what we've been doing, for those of you who are listening, our sampler has 16 filters. This is our auto sampler on the large volume multipuffer, and it carries 16 filters. And so we're running 2 filters at a time to get duplicates, and approximately 8 intervals of
Or on
2
the stove.
duplicates, approximately divided throughout the survey, which
Right.
the full survey, basing it roughly on waypoints, but
Right.
or just changes that are obvious as we move along. So, which we make a note of.
Yeah.
So,
You not bring
yeah,
give
so
us an overall picture.
roughly, yeah, an eighth of this of our 12-hour survey,
Great.
right?
Awesome.
So, but this will be a specific
You want to tilt me up to the bulb?
sample
Copilot?
that we can tie to this
What would you like?
spot.
Yeah, tilt the camera.
Awesome.
Yeah, tilt it up.
Yeah, go to the
Yeah,
bulb.
that's awesome that we have the
Yeah.
capability
I
of
think you are
doing both of
retracted
those
in here.
methods
Absolutely.
of collecting eDNA.
They're
Send
very
you back up.
complementary, yeah, for sure.
Get real close. You want to up more?
Yeah, tilt up to the top of it. Oh, I'm slipping.
Oh, sorry.
Cool.
Yeah. So in the
This is
foreground we have a
bolosoma.
sponge bolosoma. Yep. And in the background too, there's another one.
Looks like something under the oh.
Yeah.
Huh.
I have to ask, but that coral in the background in the hole, what do you think?
Ah.
If you're asking about sampling
Yeah.
it, there's no way.
Okay.
It's like
I have to
you know what
ask.
I mean?
It's my job.
Yeah, no.
It's
This is
literally
like
my job
one of the best
Come
views
on,
I could get
Levi.
at. It's like a ravine.
I know. Okay. Well, just to note that. So that's a genus called Cibogorgia.
Let's put that, and
I
maybe
can
we
try
can
and
relate
zoom
it
in
to the
that.
eDNA
Yeah.
sample.
Past
How's that?
Yeah.
I came down here
Could
to
you
eat
add
just
that to
even
the
get a
data
look
logger,
at
the
it.
name?
Yeah.
Yeah, how do you spell it?
Yeah.
I'm typing it in my notes
I'll write
as well.
it in the chat, and
You want to zoom in on it, you can.
There's something
try to-
else all the way in the back there also.
Yeah, kind of on the right, it
think
looks like
it's
something.
maybe
is
another
wrong.
crinoid?
No, it's sort of up and to the right. Up and to the right.
Oh, we're good there.
Hmm.
Look at this coral first.
so this is actually it's a type of bubblegum coral, but
Nice. Wow, that's beautiful.
oh, actually, hold on. So, I changed my mind. Is that full zoom?
That is full zoom.
So I can't be certain, but it's definitely in the Coralliidae family. It's possibly Cibogorgia, but it could also possibly be Corallium. I wouldn't be able to tell. I wouldn't be able to tell without some extra zoom, and I still probably wouldn't be able to tell if we had better zoom. So I think we'll call it Coralliidae, Cibogorgia, or Corallium, and that's the best we're gonna do.
Do you see those two little antennae things? Is
Yeah.
that coming from something else?
I don't know. Yeah, I don't know.
the
Yeah,
white-
I see
the
what you're
white-
saying.
The white sticks with curly
like
cues.
somebody-
So
Like someone's under there.
Yeah, it does make you.
I'm gonna change my mind. I'm gonna suggest it's not Cibogorgia and go to Corallium instead,
Huh.
which is
You zoom
another
out.
precious
Kinda
coral genus
slide
that's
around here.
related to the pink one we just
Yeah.
saw a little bit while ago. So I'm gonna say Corallium for the
Thanks,
genus.
video.
And it'll have to remain a mystery. Sorry.
So Steve, here's some more questions for you. How well are these precious corals represented in the reference library genetic libraries, so that when we have the eDNA, we can match our sequences to it, and
So-
maybe.
Yeah,
Oh,
I've
that's
I've
great, yeah.
sequenced both Corallium from this depth and Cibogorgia, so I have representatives
Okay.
of those two genera.
So maybe at some point we can go back and
Yep.
and see.
Yeah, that's the idea. We wanted in our initial deepwater coral reference library, wanted to sequence a broad range of octocoral genera, and these two were among them. So we will have representation of this genus in the library.
That's excellent.
Maybe the eDNA
That's why we do
will
it.
tell us a little bit more.
Yep.
That's why we do it, right?
So the difference that I would have to look for between those two genera is
Oh, what is that?
it's morphological. You can determine it with morphology. The difference is Corallium is a hard, stony octocoral, and it's fairly rigid.
All right, video. I'm going for the ball with the little things coming off it.
And
Little, yeah, I'm gonna get
Oh,
closer.
is that a
Paragorgia and Cibogorgia have a porous axis, and they're flexible.
You want to tilt me down a little bit?
Can you guess by the habitat being kind
Can I
of
zoom
under
in?
this overhang crevice area? Are there species that like that better than others?
Corallium, yeah.
Corallium.
yeah.
Okay.
That's why I'm thinking that.
Yeah.
Yeah.
Makes sense. And here's what was poking out.
Oh yeah. This is
Oh, wow.
it's called a deep sea long spine sea urchin, and the genus is
Spine sea
So
urchin.
cool.
Aspido diadema is the genus.
Spicodiadema.
And
That's
do these
I'm
awesome.
gonna wait
ones,
for that one to be spelled too.
There you go.
Steve, do these ones move fast?
They don't move very fast.
Okay.
No. So if you're familiar with long
Got more
spine
zoom? Is that
urchins,
it?
if you ever snorkeled around
Oh,
in
that's it.
coral
Okay.
reefs and you see
Thanks.
diadematid or diadema urchins, this is a relative of that, but a deep sea representative. They're primarily deposit feeders,
Yeah.
so feeding on sediments and maybe small invertebrates, but
Yeah. Clearly trying to deflect predators, I
Yeah.
think.
but they're very nice. We see them often. They're pretty
Yep.
common.
So would
Copy.
these be poisonous as well?
Or
I don't know. You
venomous.
want to try? I don't. No, they're probably they may be
You can zoom
venomous.
out video. I'm
They
moving.
may not be venomous.
Yeah, sorry.
I'm not sure, but certainly doesn't feel pleasant.
So that one rock has quite a few.
Right.
That white.
Yeah,
What an amazing
phyla.
habitat.
Yeah, really, really nice. Nice spot.
You want to snap on this white thing, see if there's interest? Yeah, that one.
Oh, yeah, there you go. I think this is, oh, what is that?
Branching looks different than what I've seen today. Oh,
Yeah.
there's something else on the rock there on the right laser.
We're going to want to collect that.
The coral.